we actually see both forms of glucose, the chain form and the ring form, but it's mostly in this form here, the ring form. Okay, so it's something like, I think it's something like 70% of the glucose is in this form, 30% is in that form. And then with sucrose, note that the form of glucose in sucrose is the ring form. And then for fructose, we see the ring form of fructose. Okay, so where is the glycosidic bond in sucrose? Okay, now let's go back one slide. We said that the glycosidic bond is the bond that joins the those simple carbohydrates together. Okay, so go back here. The two simple carbohydrates and the two monosaccharides in sucrose are? Yes, you have glucose and fructose. So what bond connects the two monosaccharides together? Okay, because don't we have a whole bunch of alcohol groups here? So here's the glucose and there's the fructose. So it looks like this is that's the bond.
The ether.
That connects. Yes, that would be the ether function. Okay, so there's our glycosidic bond. That's the ether function. So the spit in our tongue, with an enzyme amylase, that breaks that bond. It actually breaks it right there. This oxygen carbon bond right there. Okay, so once it breaks, then our body can use the glucose as a fuel. It actually turns out that our body also converts fructose to glucose, which is another enzyme which accelerates that reaction. Okay, let's move on to the next slide. Okay, so protein shape. So folding determines its activity and function. So there's a website which
I hate this diagram.
describes protein shape, folding, which structural level, primary, secondary, tertiary, quaternary, determines a protein's activity and function. Okay, so now some of you taking biology. So you take a look at that website first before answering that question?
Yes.
Or you guys know that? Know all this stuff?
You should look at the website.
So take a look at the website. Protein, what should we call it? Call it structural levels of. Okay, so here. Okay, so there are different levels of protein structure. So it says here, the wide variety of three-dimensional protein structure corresponds to the diversity of functions proteins fulfill. So it says here that the primary structure is a sequence of residues in the polypeptide chain. So what does that mean? Sequence of residues in the polypeptide chain. So For example, when I think of residue, I think of something that someone left behind, you know, like my dog, you know, goes to the bathroom with me, he or she uses residue. So is that the same type of residue? So what are the residues in this polypeptide chain? Okay, now, a protein is a polymer, and the monomeric unit of a protein is an amino acid. So actually, the residues, these are the amino acids. Okay, so the primary structure is the sequence of amino acids in the polypeptide chain. The polypeptide, poly means many, peptide is a chain of amino acids. Any polypeptide, that's another word for protein. Okay, so again, the primary structure is the sequence or the order in which those amino acids connect together in a protein. Secondary structure is a local, regularly occurring structure in proteins and is mainly formed from hydrogen bonds between backbone atoms. So-called random loops, coils, or turns don't have a stable secondary structure. There are two types of stable secondary structures, alpha helices and beta sheets. So here's an example of an alpha helix. If you can, well, it's sort of hard to tell here. What we have to do is we have to figure out where the amino acids are and where the chains connect together. So it looks like the amino acids are right here. So here's the main chain, right in there. And then, for example, see, you sort of made up that dashed line here. So that dashed line represents the hydrogen bond between this oxygen and this hydrogen between two different amino acids. So there's another hydrogen bond there. There's a third hydrogen bond. So that would be an example of an alpha helix. Note that an alpha helix, a beta pleated sheet, that's the specific type of folding within a protein molecule. Now, further, here's an example of beta pleated sheet. The tertiary structure describes the packing of alpha helices, beta pleated sheets, and random coils with respect to each other on the level of one whole polypeptide chain. So in other words, tertiary structure describes the folding of a protein molecule. Okay, so here's where they show the ribbons. So note that this, it's hard to see here, but note that this little coiled ribbon, that would be an example of an alpha helix, and then these, the sort of yellow colored thing, that's an example of a beta sheet. And then in terms of classifying proteins, there's two ways to classify proteins. That is protein folding in regard to its tertiary structure. So one is called fibrous proteins, and the other type is called fibrous. Starts with a G. And globular. Okay, so those are two types, fibrous and globular. Okay, that's the tertiary structure. Then, let's see, they talk about the ternary structure. So it says if there's more than one polypeptide chain, in other words, if there's more than one protein molecule in a complex protein, the folding of those two or more protein molecules, that's called the quaternary structure. So one example of a protein which has two or more protein molecules within it is hemoglobin. Hemoglobin actually has 4 protein molecules within that big protein, within that big complex protein. Okay, so let's go back here. And let's talk about this one here. So which structural level determines its activity and function? We thought that this study. So if you go here, the tertiary structure talks about that. Secondary structure talks about that. And they're talking about the activity and function and how it's related to structure. You always need to take a look at that website. Just in case. Let's take a look at this one. Do you guys use Wikipedia? Is Wikipedia a trustworthy website to look at?
It's okay.
Yeah,
It's
it's
okay.
start.
Yeah, it's a good place to
Yes, yeah, one thing about Wikipedia, I actually use it quite often, but I never see, because who contributes to they put all these references in, and they never say who wrote this thing. So, okay, so let's see. We talked about this activity and function here, so let's see here. Okay, proteins are a fourth class of biomolecules. Okay, so to be able to perform their biological function, proteins fold into one or more specific spatial conformations driven by a number of non-covalent interactions, such as hydrogen bonding, ionic interactions, van der Waals forces, and hydrophobic patterns. Is there an easier way to write this?
What's a van der Waals force?
That's actually, van der Waals forces, that's actually the London forces and dipole-dipole forces. So you can actually type that in like that. To understand the functions of proteins at a molecular level, it's necessary to determine their three-dimensional structure. This is the topic of the scientific field of structural biology, which is one of the techniques that's next to dual polarization interferometry to determine the structure of proteins. So it looks like, so based on this information here, is it the primary structure which determines the activity and function of a protein? So it's partially because the primary structure is going to determine the secondary and tertiary structures. Okay, because it's the hydrogen bonds between those amino acid residues which determines how a protein folds. Is it going to be the secondary structure, for example, the alpha helix or beta sheets, which determines how a protein behaves, or how a protein works or functions? However,
The way that it's folded determines how it works. So then the determining factor will be the primary structure of the.
yeah, the primary structure determines how it folds, but then it's the actual folding which is involved in the function. So, for example, one way that protein works, and this is what you're going to look at in lab today.